Okay, hello students. In this video, I am going to discuss the cell cycle chapter. I will show you that how the cell undergoes a cycle and then ultimate the division of the cell that takes place. Okay, so this cell cycle is found in every living organism where we know that living organisms are made up of cells, and where cell is there, the cell must undergo this kind of cycle. Okay, so the cell cycle, first of all, I am going to make you understand the whole process of cell cycle and cell division through a flow chart in a proper sequence. Okay, to understand the sequence is very important. So first of all, if I divide the cell cycle into two broad categories, two broad divisions or phases. So number one is the interphase. Okay, fine, let's not divide this area. We will first see the interphase only, then we will see what happens after interphase, because in the cell cycle the first step comes of interphase. Let us understand this, let us clear the concept of interphase, then we will see what happens in the next. In the interphase we see three steps that takes place or process a cell undergoes. G1, S and G2 phase. G for gap phase, gap phase 1, S for synthesis and G2 for gap phase 2. Now interphase. Let us understand what this phase all about. See, when a cell divides, before the division of the cell, it needs to prepare itself for division. There is a particular preparation, a lot of preparation is required before the cell divides into daughter cells. What kind of preparations are these? See, for example, it's a cell here, it's a nucleus. Now the cell is going to divide, okay, into two, having nucleus. Now see, the newly formed daughter cells have two nuclei, each carrying one nucleus, and also all the cell organelles present, which were also present in the parent. It means until and unless the cell organelles present in the parent cell increase in number, until and unless the nucleus present in the parent cell increase in number, it would not be possible for the parent cell to equally divide the nucleus and other organelles in the two newly formed daughter cells. So whatever content present inside the parent cell needs to be doubled. If it is two cell, needs to be doubled. One nucleus, it has to be two first. Then only one can be equally divided. I hope it is understood. Very common thing. Mitochondria, ribosome, whatever are present is the chloroplast, whatever are present inside it, the plastids and all, they need to be increased in number, so that equally can be divided into the two newly formed daughter cells. This is the preparation I am talking about. That takes place in the interphase. So basically, cell is not undergoing division. We don't say any division in the interphase. What do we see? We see preparation for division. So that's why sometimes it is also, this phase is also known as resting phase. Okay, resting phase in the point of view that the cell is not undergoing division. That's why it is resting phase. But technically it is absolutely not a resting phase. It is the most metabolically active phase. Understood? It is the most metabolically active phase. Now, most of the time of the majority of the time of the cell cycle, okay, it is spent in the interface. If a cell divides, if a cell takes 24 hours for division divide itself, including the whole cell cycle, then out of 24 hours, majority of the time is invested for preparing the cell for division, that is interface, and very less time is taken to divide only. The result, the consequence is the division, that takes very small time. The majority of the time is invested to prepare the cell for division. The interface takes the majority time. So it is absolutely not a resting phase. It is the most metabolically active phase that takes place here. Okay. Now let's see what exactly happened in these phases which comes under the interface. Okay. See, in gap phase 1, the basic points I am writing, what exactly happens here. The RNA synthesized. The RNA gets synthesized here. Number 2, the proteins also get synthesized here. Okay, and most of the cell organelles also get increases here, also get increased in number. Mainly if I talk about the membrane bound cell organelles, mitochondria, okay, Golgi complex, then plastids. Okay, so these are some of the membrane bound organelles. So they undergo this process and they increased in number. Okay, so including all the membrane bound structures, the majority of the cell organelle they increase in number. So at the end of G1 phase, what we supposed to see? We supposed to see that the volume of the cell increases because some of the substances present here, including most of the organelles present in the cell, parent cell, already increased in number. Okay. Now, there is a twist here. Before entering into the S phase from G1, there is another phase is there, which is known as G0 phase. Okay. Now, what is this G0 phase? A very important concept this is. See, we all know from our childhood days that cells divide continuously on regular basis in our body, all the cells. Now, there is little modification in the information. I am not saying the information is wrong, I am just modifying it. Remember, all the cells, there are some cells which do not undergo division on regular basis. Okay, they do not undergo. They will undergo division whenever they will be called for to do so. Otherwise, they will not undergo division. So, first of all, let us see what are those cells. Then we will see when they will be called for to do so, to undergo division. The cells, for example, our neurons, our nerve cells, our brain cells, the liver cells, the cardiac cells of our body, these are the cells which do not undergo division on regular basis. Now question comes, why? Skin cells undergoing division regular basis, no problem at all. Why brain cells cannot undergo division regular basis? Why liver cells are not doing division? Why cardiac cells are not frequently undergoing division? See, remember one thing, at a time cell can do only one function. Either it will divide or it will perform function. Both together cannot possible. Dividing and also performing function simultaneously, it is not possible. Either it will divide first, then it will perform function. Okay, so one function at a time. So that is why now see, if neurons undergo division on a regular basis in our brain, it means those neurons for some time in a day do not perform function. So what do you think? Is it a natural phenomena happens in our body? No. Then part of my brain cells on a regular basis, okay, will undergoing division. So it will not be functioning. That part of the brain will not be performing any function. So everything will stop. It is really not possible. My brain has to function 24 hours. When we sleep also, then also our brain cells, our neurons perform function. They cannot stop performing function. It means if they cannot stop performing function, they cannot undergo division on regular basis. Our liver cells also perform very important function. Our cardiac cells also perform very important functions 24 hours. Our heart beats 24 hours. It cannot stop. Once it stops, we will stop. Our life will stop. Okay, so these are the cells. They are not supposed to undergo cell division on regular basis. This kind of cells, when they come in the G1 phase, they escape the phase and they enter into the G0 phase. They do not undergo S phase. Okay, they undergo directly from G1, they undergo the G0 phase, which is actually a phase where these cells undergo and they are there and they are metabolically active, means they are performing function. When they undergo G0 phase, they are normally perform their function, not undergoing division. Understood? Now apart from this, there are cells, sometimes it happens that cells undergo, normal cells, our body cells, not brain cells, I'm not talking about brain cells and all, any cell that undergo G0 phase sometime. Why? What about those cells? See, sometimes what happens, if there is lack of nutrients in the body, lack of sufficient nutrients in the body, okay, it may happen that for some time the cell undergo this G0 phase, and whenever the particular nutrient is availed in the body, the cell will get sufficient nutrients available in the body, again they will undergo the further divisions or further process of cell division. So this is the one reason that cells undergo G0 phase. Okay, 2 point of view I have discussed with you, that cells undergo G0 phase, when and why and which are those cells. Now after, so normally what happens after G1 phase, we will see the GS phase. Now normal cell undergo this direction. Okay, so in the S phase what happens? S means synthesis phase. What happens here? DNA replication takes place here. Very important function. We know that RNA, DNA, these are the genetic substances, genetic materials. They carry the information, genetic informations. Very important. Okay. So DNA replication takes place here. It means that DNA number gets doubled. Along with it, what happens? Chromosomal duplication takes place here. Chromosomal duplication also takes place here. Okay. What is chromosomal duplication? Means, is there a difference between replication and duplication? Yes, there is difference. DNA replication is number of DNA increases. Chromosome duplication means the chromatids present in the chromatid present in the chromosome, they get doubled. Means normally when a cell is not undergoing division, its chromosome means one chromatid. But now what happens during duplication, that chromatid duplicated and it forms two chromatids. Okay, so chromatid number increases. So before cell undergoing this process, one chromosome means this, one chromatid having DNA. Once duplicated, the DNA number increases and automatically chromosome duplication means the chromatid doubled. Now the chromosome containing consists of two chromatids. So from here we will say the chromosome contains two chromatids, not one chromatid. Okay, and they are connected by centromeric junction connection. Okay, so chromatid number increases but chromosome number remains same. Remember this. Okay, now what else happened? Centrioles also replicated. So from 1 to 2 centrioles come into existence here. Clear? This point is very important, remember, because this information is going to help you further in the next steps. Chromatid number doubled but chromosome number remains same. Let's come to the G2 phase. G2 phase is a repetition of the G1 phase normally. So RNA synthesis continues, protein synthesis also continued, and whatever the cell organelles present there, the cell organelles increased in number. Done? So these are the primary, the functions, the main functions that is done, okay, during the interphase in this, under these phases or stages. So we have completed the G2 phase. Now something very important I am going to tell you. There is a concept called checkpoints. Okay, there are checkpoints, at least 3 checkpoints are present in a whole cell cycle. Within the interphase already we have crossed 2 checkpoints. Okay, one checkpoint is after G1 phase. There is a checkpoint. After G2 phase, once it completed, there is a checkpoint. 2 checkpoints are there. Okay, so what is checked here? It is checked first of all in G1 phase. It is checked whether RNA or protein synthesized properly or not. After G2 phase, it checks whether DNA replicated properly or not, whether chromatids increase in number or not. What is the number of chromosomes now? Everything is checked in these checkpoints. Okay, if any lacking found, it stopped there for the time being. Like some cells, okay, it has been checked here that some cells are lacking nutrients, and nutrients are required by the cells for the further divisions, because this is cell division is a heavy process. It needs energy. So cell needs nutrients. In this checkpoint, suppose it has been found that some cells are lacking nutrients, directly those cells will be sent to the G0 phase for the time being. Once the nutrients get are available in the cell, again they will coming for the S phase and G2 phase. So these are the checkpoints. Okay, whenever any problem will be found for that particular cell, the cell division, the process will be stopped there, pause there for the time being. Again, when everything becomes normal, things are available, then again it will move on. This is the function of checkpoints here. Okay, now after G2 phase, the cell is ready for division. Now we will see what happens after interphase. Interphase is done. Okay, after interphase, see, we have 2 types of cells in the body. Okay, one is somatic cells and another is reproductive cells in our body. After both these cells undergo interphase, so both of them they crossed interphase already. Okay, for somatic cells what happens? Somatic cells undergo M phase, that is mitotic phase. and reproductive cells undergo meiosis cell division. Under mitosis cell division, there are two steps. One is karyokinesis, another is cytokinesis. Nuclear division and cytoplasm division. Under nuclear division, what states we see here? Prophase 1, metaphase 1, anaphase 1 and telophase 1. 1. Prophase, metaphase, anaphase, telophase. These steps in this proper sequence it takes place. Okay. So now by the end of telophase already the nuclear division completed, and then from here it will undergo the cytoplasmic division. The cell will divide. Okay. In meiosis same, it will be a karyokinesis phase, nuclear division and cytokinesis. But in meiosis, remember, the karyokinesis, the nuclear division takes place in two different stages. One is meiosis 1, another is meiosis 2. Why? I will discuss. Meiosis 1 having prophase 1, metaphase 1, anaphase 1, telophase 1. While M2, meiosis 2 having prophase 2, metaphase 2, anaphase 2 and telophase 2. Clear? Once it does, here telophase 2 it is done, so nuclear division of these cells are done. After this it will move to the cytokinesis process. Okay, so this is how these are the different cells, they undergo the process of division. Clear? So let me first tell you the basic differences between mitosis and meiosis. Mitosis takes place in the somatic cells, the normal cells of our body, and meiosis takes place in the reproductive cells present in our reproductive system. In mitosis, one cell divides into two new daughter cells. At the end of telophase, cytokinesis means mitotic phase, we will see two new daughter cells are produced. In meiosis, 4 daughter cells produced. After meiosis 1, 2 cells come. From one cell, 2 cells come after meiosis. And after these cells again undergo further division, and 2 cells will be produced. 1, 2, 3, 4. So after meiosis 2, these 2 cells undergo meiosis 2. So it produces 1, 2, 3, 4. 4 cells are produced. Daughter cells. Okay? So meiosis 1 happens here, meiosis 2 happens here. Third most important difference. In mitosis, the chromosome number remains same. What was happening here? One cell divides into only 2 daughter cells. If it is 2n, it is also 2n, it is also 2n. So chromosome number remains same, equational division. In meiosis, it is called reductional division, chromosome number reduced to half. If the parent is diploid cell here, after the meiosis 1 only it will become half, n, haploid. Then these 2 cells undergo meiosis 2, chromosome number remains same here. For these cells, daughter cells, this is the parent cells. So here the number of chromosome will remain same. They were haploid, they are also haploid. But here we see the reductional division. That's why meiosis is also known as reductional division. The chromosome number reduced to half. Okay. So in this video, this much only. In my next video, I am going to discuss these processes, whatever I have shown you here. Okay. The mitosis and the meiosis detailed structure, detailed explanation of the process mechanism, I will discuss in my next video. I hope the basic overview I have already given to you. Okay, please note down, make a note on your own. So it is very important to learn to make notes. If you want, after making notes, you can also send me some of you, I can check also. no problem at all. So make your notes. Okay, pause my video, note down again, restart, pause. By this process, note down all important key terms and make your own note. This is going to help you a lot. Okay, thank you so much for watching this video. Wait for the next video where I'll be explaining the mitosis and meiosis in details. Okay, thank you.